This is the Pacific oyster. Scientific name is Crassostra gigas. It was a Japanese oyster that was brought into the state of Alaska in the early 1900s. It's the farm product now that we have in the state of Alaska for the seafood market. It's a cupped oyster because, as you can see, the top of this shell is cup-shaped and the other side is flat. When grown in the wild, they cement on hard rock beaches, hard surfaces, with the cup side down like this, and the top exposed. The outside of the oyster shell is called the peristrachium. It erodes away when the shell is very small. In the adult, the peristrachium is actually gone. This particular layer is the nacre layer, or the next layer down. It's got an interesting color pattern. It's a little bit white, a little bit red, and a little bit darker brown and black. The color of the oyster shell is actually a genetically programmed process in which a shell like this is a combination of a darker shell breeding with another oyster that has a white shell to produce this mottled type of coloration. When we shuck an oyster and open it up, we actually, in Alaska, shuck it from the hinge on the pointed end of the shell. If we try to break through any other part of this shell, it fragments so badly that it makes the oyster difficult to eat. So we shuck the oysters from the hinge. When I want to shuck the oyster here, I'll insert the shucking knife into the hinge here, twist it until I feel it loosen up slightly, and then pry it open. Here, I'll poke it in here. That hinge is now broken, and we're ready to open up the shellfish. The issue here now is there's a muscle right in the middle here that holds this shell shut, so I have to insert this knife in here and sever that muscle. Once severed, the shell can then be removed pretty easily by just cutting around the edge and removing it. It was just harvested, so you get a lot of water. And then initially when they come out, they'll dehydrate in time if they're left out of the water for any length of time. Here's an example of a dry oyster. You don't want to eat this. This is going to be a poor quality animal because it's just a lot of sediment in here that was not flushed out. In the oyster here, there are a number of structures that have biological significance. The mantle has composed of two particular parts. One of them is the lip of the mantle. As you can see, this dark structure. If you turn this over, you'll see that there's a membrane that goes across the entire surface of the mantle. This is the membrane of the mantle. The lip here and the membrane. The lip of the mantle is responsible for the actual growing of the shell, putting the calcium carbonate and depositing it down along the shell. The membrane of the mantle is responsible for the thickness of the shell as this oyster grows. So both parts of the mantle have that function. In order for an oyster to form the shell, the calcium carbonate in that shell, the concentration of the calcium carbonate needs to be greater than that of the seawater. In order for that to happen, this mantle is sealed around here, and when it's sealed against the shell, it forms a chamber between the membrane of the mantle and the surface of the shell. That's called the pileal chamber. And what happens is that the calcium carbonate that's in the seawater actually gets concentrated through the membrane of the mantle, creating a concentrated solution on the other side. If we look at the other parts of this oyster, is actually composed of a number of structures. The reproductive structures in here. All oysters are males in their first year in Pacific oysters, and they turn to females after the age of 2. And so they split apart into male and female. Generally speaking, if the environmental conditions are good and healthy, you'll get more females than males. If the environment gets very lean and the food is not as abundant as it should be, there'll be more males. This dark structure in here is what's called the digestive gland. It is the portion of the digestive system that breaks down the food that this oyster consumes. Oysters actually bring in food from a current flow that's generated by the oyster. And that current flow is generated by this structure here. This is actually the gills of the oyster where they extract oxygen and exhale or get rid of the carbon dioxide that's in their system. So this is the gills here. Very fine filamented gills that have very fine filaments that go out from the middle of where the gill is attached. On the surface of these gills, there are very, very fine hairs, tenidia they're called, and as they beat, they create a current that moves the food into the oyster, into the esophagus here, eventually through the digestive gland, and the digestive gland and the intestines are wound within this mass of ovary or testes, also the digestive gland and the remainder of the digestive system. And so that's what happens with the food. It comes in and into here and back out again. The oysters use this hair-like filaments that are beating as sorting mechanisms to sort out particle sizes. So if the particle is too big, let's say like a grain of sand, for example, it will not make it through this filtration system and be expelled. If you get very large algae, microalgae out there that the oysters consume, if it's too large for them to feed on, they will expel it. This whole process of filtration is really quite profound because if you have high sediment waters that create a real large load of organic or solid materials that are too large to be filtered through here, the oyster is expelling a lot of this material from the inside through this filtration system. It actually reduces the feeding rate. That's why oysters, when they are in the water, grow better where the water is not so turbid and muddy and full of particles, because they're not wasting energy and the efficiencies that are used to get the food they need. So clean water is important. As we go into them internally, this is the adductor muscle. Remember the muscle we cut earlier? The adductor muscle is actually the two parts here. One of them is the white muscle, and this is the striated muscle. The darker striated muscle is used to open and close the shell. So if you've got the shell on like this, if you're opening up, it's relaxing the adductor muscle. When it's contracting, it closes the shell shut. The dark muscle is interesting because it is used to open and close the shell, and it's also a muscle that requires oxygen to do that, due to the kind of contraction. But also the striated muscle can get exhausted quickly because it uses oxygen and it exhausts. That's where the white smooth muscle comes in. It's called the catch muscle. It's an interesting function of this muscle because the striated muscle closes the shell and the catch muscle locks it. The catch muscle doesn't rely on oxygen. When it locks, it locks. So it can stay that way for an extended period of time. It lengthens the time in which that shell can be shut. Now this makes sense for an oyster like the Pacific oyster, which is intertidal. It goes dry when the tide goes out, and so it's exposed to air. Shell gets shut, the catch muscle locks it, and it stays shut while the tide's out. If you are buying oysters, when you go to the store, you look inside the refrigeration system. If all the shells are really tight closed, and it's in good shape. The general rule is you don't want to buy an oyster when it's gaped open. A gaped oyster would be with a shell opened like this, because that could mean that that's been out of the water for an extended period of time. Now what you would like to do with this thing, if you could, is to thump it with your finger, like that. If it's snapped shut, you know that, oh, that's pretty good shape. If it doesn't close, put it back. It's not something you want to buy. You run the risk with your personal health by consuming something like that. As we work our way to the back side of this tissue mass, you'll see the mantle, and you'll also see various types of colorations of the calcium carbonate. Calcium carbonate shell looks mottled. You've got dark spots, you've got light spots, and you've got what's called chambering happening in here. Now this oyster has gone through some chambering. What this indicates, and it's not uncommon to have this happen in Alaska, to have these softer calcium carbonate deposits. In the northern climate like we have in Alaska, the summer is a time for abundant growth. Lots of food, water temperature warms, they start growing very well. Then as the winter comes in, as the animals get skinnier, and the shell forming process gets altered because there's so much energy that's needed for shell formation and there's not enough nutrients to go around, that you'll get some of these softer deposits. So when you're shucking the shells, you can eliminate that potential of getting a lot of that calcium carbonate by breaking the hinge and severing the adductor muscle. This inner layer of the shell is called the mother of pearl. The mother of pearl is oftentimes termed as a place where the pearls get formed. And I often get questions about, do these Pacific oysters form pearls? Well, yes, they do, but there's not much to them. They do not form the classic mother of pearl, jewel quality pearls that are available for the market.
We'll get back and finish up. Did you want to get that back?
We can go and have another. Learn the Yoga Sutras. That's about my level.
Patanjali's aphorisms, or
Okay, we're leaving now.